This conference will now be recorded.
Welcome, everybody. Just a reminder for all the audience members to please go ahead and mute your microphones for today so we don't hear any unanticipated background noises. My name is Dr. Mary Beth Neely. I'm GEO AquaWatch's scientific coordinator. It's my pleasure today to introduce our webinar presenter, Loé Maire from EAWAG, which is based in Switzerland. And she will be presenting her doctoral work or some of her doctoral research on differentiating phytoplankton taxa in subalpine lakes. And she'll define what all of that means to us in the audience coming up soon. And just a reminder, this recording today will be available a little bit later in the afternoon. It will be posted on our website. And in case you missed it or your colleagues miss it, you can rewatch it there. We will be moderating any questions in the chat. And you can also unmute at the conclusion of Loé's talk today and ask your questions of her directly if you like. That's up to you. And I wanted to also mention we have recordings of our past GEO AquaWatch presentations also available on our YouTube channel. We do have one more webinar coming up in the series. It will be next month in May for this academic year. If you're interested in presenting, go ahead and I'll put that information about that upcoming webinar in the chat. If you're interested in giving a webinar yourself about your own project on remote sensing and water quality, you can reach out to me and we'll make arrangements to get you scheduled. So at this point, I would like to introduce our speaker. Loé Maire, as I mentioned, is currently working at EAWAG. She's also a remote sensing laboratory PhD candidate at the University of Zurich. She already holds a Bachelor of Science and Master of Science in Environmental Studies and Engineering from École Polytechnique Fédérale de Lausanne in Switzerland. And I'm sure that I mispronounced that horribly, French not being my my first language. So apologies to those in the audience. You can eat a cookie and hopefully feel better about that. So go ahead and take it away, Loé. Welcome. Thank you.
Thank you very much, Mary, for this nice introduction, and thank you everybody for being here today. So as Mary said, so I'm now doing a PhD at University of Zurich and also at EAWAG, and I will present you now a first part of my PhD work. So in our group at EAWAG, we are studying surface waters, mostly lakes, and in Switzerland with remote sensing. So in Switzerland, we have around 1,500 lakes going for a few hundred meters up to 5 to 6 kilometer, 100 kilometers square. So we have a large diversity of lakes. So we mostly focus on Swiss lakes. And in some of them, like you can see here on the picture, we have some research platform which provides us a lot of in situ data, which will be here on the heart of my talk. That's why I here have a picture. So indeed, my talk is about how to differentiate the phytoplankton taxa in lakes using these in situ hyperspectral reference data. So in Switzerland, we actually also witness some algae blooms, some phytoplankton blooms that we can also witness usually also in oceans or in coastal waters. In Switzerland, it happens quite often, at least once per year, that it blooms to a certain point that we have news in the newspaper, article in the newspaper, to care to so that the population is have to be careful about this, because people used to swim or have some activities around the lake. And these are, for example, of 4 different years, or in 3 different lakes in Switzerland, where we had like really large and problematic blooms in lakes. So it's a really well-known problematics here that we study a lot. And these phytoplanktons also show is also a witness of the human activities along the lakes. For example, if there's a lot of farming or so, but it's also witness the change of climate and the warming of the temperature. So this is a problematic that we study a lot. However, phytoplanktons, when they don't bloom, they are actually quite positive for the environment, because they are at the base of the aquatic food web, and they are really important for the carbon pump. So they will usually capture the CO2 and free some O2. So they will perform photosynthesis and carbon sequestration. Then they will be eaten by small fish and then big fish, and then they will sink to the sediment when they will die. So they will sequestrated carbon, which is also a good thing. And then all of the different taxa also play specific role in the biogeochemical function. So some of them are nitrogen fixers, some calcifiers, some silicifier, and so on. So they all have a really different and specific role in the carbon pump, in the ecological pump, and they also witness the climate change. So for all of these reasons, it's really important to map phytoplankton diversity. To do so, you could go to a lake with a boat, take a sample, and then come back to the lab, and then do the measurement of phytoplanktons in the lab. However, this is really first time consuming. Then you go to only one place or to only one lake, and then it's really repetitive, and then also it's cost a bit. So to do this a bit more easier, and also to have a higher spatial resolution and higher revisit frequency also, we would like to map it, this phytoplankton diversity from space. Until now, the remote sensing studies about lakes is really underrepresented until actually the last few years, because ocean has been studied a lot, because they play a huge role in the Earth system. But now we also understand that lakes are quite important at the local scale, so we start to study them. But also lakes are much more complex than ocean, for example, because we have to take care to take it into account the non-algal particle and the colored dissolved organic matter. So we have to take it into account some particles in the lake that also play a role in the optical patterns. And until recently, all the satellites that were developed were more developed for terrestrial problematics or for oceans one, and in the multispectral satellites world, there is no one specific for inland water. However, now we start to see that those new hyperspectral satellites and the future one, so the new like Prisma and MAP and Space, and the future one like Flex, have actually a lot of potential to study inland waters. So in this research, we would like first to study inland water with in situ data to develop a method. And then when we would have developed the method, we could apply it to satellite data. So the method is to distinguish phytoplankton taxa with hyperspectral in situ data, and then use an inversion model for this. So our study area is a lake in Switzerland. So this is the map of Switzerland, with Zurich being here, where I'm now studying. And next to it, we have a small lake, Greifensee, which is around 3 kilometers by 1 kilometer, so rather small. And it was highly hypertrophic in the 1970s, also because there was a lot of, there is still a lot of farms around. However, in the late 90s, they had some regulation about phosphorus, so now it's still hypertrophic, but not hypertrophic as it was before. And on this lake, we have an automated research platform, as I show you in the very first slide. And in this platform, we have a lot of monitoring instruments, so we can actually monitor this lake really well. On this lake, on this platform, we have a phytoplankton camera. This camera was developed not by our remote sensing group, but by another group at EAWAG called Ecological Group. And they developed this camera, which they installed in Greifensee in 2016. And it's now since 10 years in the lake and at 3 meter depths. And it basically take picture inside the lake, 1 picture per second for 10 minutes every hour. So it's a huge data set that we have. And once they took a picture, then there's a machine learning algorithm that will classify the picture into taxa. So they will detect the region of interest. Sometimes it's phytoplankton, sometimes it's just like a detritus, for example. And then the phytoplankton, they are able to classify them as a taxa with a machine learning algorithm. So we have a huge database since 10 years of phytoplanktons in our lake. This database was used a lot in their ecological group, but in our remote sensing group is the first time we use it as a validation dataset. And however, the problem is that the units that we are studying here is area per second, so the section of the phytoplankton. At the same platform, in the same area, so this is the platform, we have a WISP station, which is a spectral radiometer that was developed by Water Insight, and we installed it in October 2020, so 6 years ago, and we have measurement every 15 minutes. And it basically measures downlink and uplink radiance, and then computes irradiance. And then with an algorithm, we have a correct equation for sun and sky glint, and then we obtain the remote sensing reflectance. So this remote sensing reflectance is my main study datasets from which I want to obtain my phytoplankton taxa. How does this work? So to obtain phytoplankton taxa from this remote sensing reflectance signal, I will use the watercolor simulator software called VASI that was developed by Peter Gege at DLR in Germany. For this, the watercolor simulator needs some reference-specific absorption spectra for each phytoplankton taxa. So I took some taxa that are commonly present in Greifensee, in my lake of interests, and I used the specific absorption spectra that were as default in VASI, but also implement new one depending on my lake. And this absorption spectra would be the base for VASI to determine the concentration of each of these 5 phytoplankton that I have here. So how does this work? You have to first take your remote sensing reflectance spectra from the WISP station, so from the field work, and then you import them into VASI. And then the VASI inversion works so that you have to do some initialization with some parameters, and then you will fix and fit some of them. You can't fit that much of the parameter, otherwise you will have a nil post problem. You will have overfitting. So we only fitted 5 phytoplankton taxa, CDOM and non-algal particle. And then you have to run inversion. When VASI will run inversion, it will actually find reflectance spectra that fits the best your reflectance spectra. And then from this, it will know the concentration that are the phytoplankton concentration using the downhill simplex algorithm. And then it will give you a phytoplankton concentration. And when we have a concentration that is below 1 milligram per cubic meter, we said that it was too low, so we put it to 0, set it as a new constant, and run again the inversion until we have fixed phytoplankton concentration above 1. And then once we have our phytoplankton concentration from this WISP station, we can compare with the abundanced phytoplankton dataset from the camera. As an example here, that's the reflectance spectra that we obtained from VASI at during different blooms. So for example, the first one is during a cryptophyta bloom, this one during a green algae bloom, this one during a cyanobacteria bloom, and this one when there was no bloom. So first you can see that the reflectance spectra is really different, and then you can see also in red that the fit from VASI really fits well the remote sensing reflectance spectra from the field in black. So to see if this works well, we first study days where we had no clouds, so cloud-free condition. As an example here, you have 2 different days, one in 2022 and one in 2023. On the left, you have the data from the camera, and on the right, the data from VASI. The data from the camera, so they are the lines, and from VASI, they are the dots. And as you can see here, the camera saw a high concentration of green algae. And then when I inverted with VASI, I could also retrieve the bloom of green algae. And then in this day, we also have from the camera a bloom of cyanobacteria, and then the dots are the data from VASI, and it also retrieved a bloom of cyanobacteria. We got some situation where the camera sees, so 2 different, so for example, 2 taxa with a really high abundance, so kind of a bloom of 2 different taxa. And in this condition, VASI can retrieve well the 2 taxa, for example, here green algae and cyanobacteria, but maybe it will mix a bit the concentration, but at least it will retrieve well the 2 taxa that are actually here. And we have one case that I will also explain a bit more after, but we when we have a cryptophyta bloom here, for example, I mean, it's not a bloom, VASI retrieves a well cryptophyta, and then he mixed it with cyanobacteria. So we also saw a lot of time where cryptophyta was mixed with cyanobacteria. But as you can see here, actually the concentration of cryptophyta here is much lower than those blooms, 3 blooms here. So that's also a point that I will mention after. So this experiment with cloud-free condition, we did it with 12 days. And we find this correlation matrix that shows that actually the results are really good for green algae, quite well for cyanobacteria and diatoms, but actually doesn't work that much for cryptophyta. So then we wanted to see if we can do the same, but under successive blooms. Meaning here, I don't do any filtration with weather. I just took in 2022 from June to October, we have like different bloom of different taxa, and I just took 1 day over 2 and inverted the day. And here, if you see, the data from the camera are in blue, and the data from VASI are in green. And the camera, for example, sees a bloom of green algae, and then a second one. And you can see in green that when I inverted with VASI can really well retrieve the concentration of green algae, the first bloom, and then the second bloom. Same for diatoms. We had a bloom in diatoms in October that VASI can retrieve really well, and even more for cyanobacteria. We have a bloom also at the end of the year that the inversion by VASI retrieves really well the increase of cyanobacteria concentration and then the decrease. It doesn't work for Cryptophyta, but as I mentioned just before, the concentration is much lower than for the other species. So here it's 10 to the power minus 11, and then it's here 10 to the power minus 10. So in our lake, Cryptophyta is always present, but as a baseline, it never blooms. So it's always difficult for us to retrieve Cryptophyta in our lake, at least for the years I studied until now. I did the same for 2023. We had now consecutive blooms from also June to a bit later, beginning of December. For green algae, the inversion also works really, really well. We have 2 blooms that Vasi inversion can retrieve really well. For diatom, we have a bloom at the end of the year that Vasi in green has a bit hard time to retrieve. At least it doesn't show, it can't see the beginning of the bloom. And for cyanobacteria, it can see really well the increase, but not really well the decrease. So I wanted to understand why, and I looked at the weather at this time. So I actually looked at the downwelling irradiance that we had at this time, and I fitted a sinus curve to this downwelling irradiance, and I look at the difference between my theoretical sinus curve of downwelling irradiance during one day and the downwelling irradiance that we obtained, we measured during one day, and I computed the loss. And what you can see here is that the higher is the loss, meaning that we had much more clouds. And the dates, like around mid-October, we have Vasi inversion that works a bit less. And it's the time at which we can start to see a lot of clouds coming. And also, we have to say that around November and so the sun zenith angle is much lower, and so than July and August, and may also increase the error. The last reason that we could have also some error in cyanobacteria or diatom is that, as I said, to do this inversion, I need some specific spectral absorption. And this spectra, I took one spectra for one taxa, but actually there's maybe much more diversity. In cyanobacteria or in diatoms, maybe they have some other species that have a spectra with a peak a bit at another wavelength also. So maybe the spectral variability inside the species is higher than what I imported now into Vasi. And we wanted also to look at the relation, because now in my data I compare the camera data that look at the area of the cells, and the Vasi, which compare the quantity of pigments in a cell. So we have actually 2 different units. So I wanted to compare this relationship that you can see here. I did separate 2022, 2023, and then combine them together. We can see that a lot of them, a lot of those correlations have p-value that is not significant, so I can't show the trends. However, I can say that here in 2022, cyanobacteria shows the best correlation, and in 2023, green algae the best correlation. And in both years, they are respectively the concentration with the phytoplankton with the highest concentration, meaning the highest the concentration, the easier it is for Vasi to retrieve them. And however, I want to say that until now I mentioned the bio-optical limitation that could give an error. For example, low concentration or a high diversity in the spectrum for the different taxa. However, I must mention that if we have an ideal scenario where we have error-free measurements, this curve here should not follow a linear relationship, but should follow a relationship which is area to volume, because we look at the area in the camera and the volume in the Vasi. And on top of that, in a phytoplankton cells, like all phytoplankton cells, all cells in one taxa may be of different size, and one cell may have a different pigment concentration. So also this curve here depends on the cell size and on the quantity of pigments in the cell. And to witness and to a bit study this, we wanted to see actually for each of those four taxa how the size variability would impact our results. So if you look here for the four taxa, you have a bar plot of the frequency of taxa per cross section, so per area. So for cyanobacteria, I cut the plot here, the x-axis here, but actually it will continue here until 2.7 to 10 to the power of -8. So it's there is actually much more cyanobacteria also present here at really low frequency. But just this is mean, like if you look at the relative standard deviation, it means that actually there is a lot of cyanobacteria with a lot of different size, and this impacts also probably our error measurements. The fact that in cyanobacteria they have a lot of species with different size. Diatoms and green algae have also moderate variation in size. You can see it really well here. It may, like this difference in size may also impact the concentration that you obtain at the end. However, for Cryptophyta, they actually have almost no variation in size. They are all really small, like between 0 and 1 here. So it means that for Cryptophyta, the size variability is not a cause for the error that we obtain. And as I said, it should be and probably be the reason is probably because its concentration was really low in our lake. So this was the last study we did here. And as a last step, I would like to say that we compare two different data sets with two different methodology, but we have a really nice agreement between both. So we can say that this spectral machine inversion with VASI is a valid approach to retrieve the phytoplankton taxa from in situ hyperspectral data, because we can actually detect different blooms. However, low concentration are a limitation. We are now today trying to understand which concentration does low mean. And then we also see that the variability of spectra inside a taxa is also another limitation. We could also think about changing the specific absorption spectra into VASI. Also, this study is constrained by the lake we choose. So it would be, now we also have some colleagues who try to do the same, but with Lake Geneva, which is south of Switzerland and which is much less eutrophic, is mesotrophic. So we want to see if it works the same with a lower concentration. And now that we also have a hyperspectral satellite mission, such as Prisma and MAP and PACE and FLEX that is coming, we would like also to test it with satellite images to see if it would work. So overall, this study is show a lot of potential to map phytoplankton diversity, to track their dynamics, and thus to improve lake ecosystem management. Yeah, if you have any question, I'm still here for quite a long time.
Wonderful. Well, while everybody is reacting and finding their applause buttons or their raise hand buttons, I'll go ahead and ask a question that I had a little bit earlier. Is there any sort of difference in your workflow if you're using this in marine waters? Well, let me back up. Is it possible to use your workflow over marine waters? Is there any change that needs to be made or no?
I would say that it should be possible. Firstly, because in marine water, I mean, there would be some change because in marine waters, I think you don't use and you don't take into account sedum and nap. So then when you will do the inversion in VASI, to my knowledge, you should only fit the phytoplanktons, the 5 phytoplankton species. So then you fit 2 parameter less. You don't fit sedum and you don't fit nap. So
Okay.
which actually really good because then you you improve the you improve the fact that you have probably less overfitting and less ill post problem. And then so I think for this it would be really easy to apply it to marine water. Yeah.
Okay, so some slight things to take into account. I'm not seeing any hands raised, but holler if you have a question. I am seeing Jean-Michel Martinez has a comment. You showed only near midday data. Are spectral data acquired earlier in the day or after 3 p.m. very noisy, and that's why you didn't include it?
Yes. So our WISP station that we can see actually here records from 8 in the morning to from sunrise to sunset. However, we did some study in our group of these data sets, and we see that from early morning and late afternoon, our data sets really show some. The quality control really shows some spikes and was not really good. So that's why we now focus. We start at like around 10 and finish at around 3 p.m. because we had we studied before with our data, and it showed that too early in the morning or too late in the afternoon, the data show really bad quality. Yeah.
Okay, great. Lots of people are expressing interest and compliments on your talk. I'll also just point out that I was very fascinated by your interest in the diatom signature. And I can tell you from my work in Florida, we always have diatoms, but sometimes it tends towards more smaller chain formers because it's a silica limiting environment. Now that wouldn't happen in your area, but I'm sure it makes a dramatic change on the signature, spectral signature. Any other questions? I'm seeing a hand. Let me see who that is. Chuan Min. Chuan Min, we aren't hearing you, so maybe you're on mute. Okay, maybe Chuan Min, you could put your question in the chat and I'll moderate it. Any other questions from the audience? Okay, let's see if Chuan Min, can you try to unmute again, maybe? Okay, well, Loé, could you please drop your email in the chat, perhaps?
Yes.
And we can get you connected with Dr. Hu from the University of South Florida, who probably has some great insight and inspiration to share with you, but maybe also a couple questions. Oh, Chuan Min is now typing his question. So, yeah, we will stand by for that. Just a reminder for everybody on the call, the recording will be posted on our YouTube channel a little later this afternoon. Could be as late as tomorrow. Sometimes it takes me a little while to get that up there. Also, our next, the details of our next webinar, which I think is on May 15th or 13th, are in the chat log as well. We will be sharing a recent outcome of a recently published workshop report that I think will be of a lot of interest to many on the call. Some of the benefits are already shared on our website, but this would be a deep dive into that. Okay, Chuanmen's question is, of all those hyperspectral wavelengths, which wavelengths are really key to your results that you presented today?
I would say that's three quick questions that I'm trying now to answer since a few weeks / months. As you maybe here, as you can see here, we have the spectra that I was interested in, and for example, for cyanobacteria, we have a huge peak here for the pigment phycoerythrin, and we have this one for the phycocyanin, and also here for the Cryptophyta, we have this peak here. So we have, I mean, it was also already kind of studied in some paper which wavelengths should be of interest for the different pigments. Now, in my study, since the past few weeks, I try to understand which exact wavelengths will be important for those species. It's actually hard to tell one number. Like this, I could not say one number. I could say some regions where we have the major peaks and the major dips would be the most important. But like this, I can't say one wavelength with one number. Yeah. And so far, what I've studied the past weeks about these questions is that the work that has been done until now by other water researcher about this topic is actually good enough to my point of view, and the result that I obtained until now is not a major advance, or doesn't complete or go against what I've been already shown. Yeah.
Okay, so I think that there is a follow-on from Chuanmen. Maybe you already addressed that. He suggests maybe there could be just 20 wavelengths that would be key to your results, to get your same results. Now, the work that you talked about maybe doing in the future, where you're looking at maybe like complementary wavelengths for diagnostics, that might be a little different. Also, if you have it handy, apparently I fixed a bad link in the meeting invitation for your paper. If you have it handy and you can drop it in the chat, that would be, otherwise I will link it more effectively underneath the like in the caption for the YouTube.
Yeah, I just would like to react to what you just said. I did perform some inversion with Olchi, so multispectral sensor, and the results were really I mean, we're not working at all, to be honest. However, probably it may be because of the placement of the bands of Olchi. This I don't know. Maybe if the bands of if you take the same number of bands as Olchi, but then you put it in different wavelengths, it would work maybe. However, I did try with Olchi, and the results were not convincing at all.
Okay, thank you for clarifying that. And then if you do have the link, go ahead and drop it in the chat to your recent paper. If not, I will also make sure if anybody had to leave that that a better link is made available online. And I would also suggest that unless it goes to some sort of random page, I probably was trying to send you good information. So you might want to read that, whatever paper I linked, you might want to also read that too, just for fun. I know we all are. Yes, it was indeed a different paper. Hopefully just as compelling though, Irene. Okay, so once again, another round of applause for our speaker today, Loé Maire from EAWAG. Good, best wishes for completing your degree. And we hope to invite you back for many activities with GEO AquaWatch in the future.
Yeah.
And I see Loé has definitely shared the link, the proper link now in the chat to her publication. So thanks everybody. And we hope to see you next month and have a great rest of your day.
Thank you everybody for being here. And thank you, Mary, for organizing all of this.
Of course, it's my pleasure. Have a good day.